Welcome everyone to this special brown bag. To say at the outset, today's lunch was donated in honour of Dorothea Endicott and her friendship with Bill Ziotis. Heard about last night, Ziotis. We're very grateful for this. There really is such things as free lunch. So we'll have to listen to it. It's a great pleasure to welcome Beatriz for many years back. Received a PhD from the University of Pittsburgh. He's currently Sport Professor of Psychiatry and Pediatrics, Professor of Psychiatry at Pittsburgh. She's also the founder and the director of the Laboratory Fellow Western Psychiatry. It's my second time of saying it. We actually first got to know each other, I think, when we study session.
Exactly.
Many more years ago than either of us would like to. But we've known each other for a while. And last night she gave a wonderful public great. So she is renowned for her research on the basis of behavior during adolescence, or to research papers on these and related fields. And among other honours, became an elected member fellow of the National Academy of Medicine last year. So please join me in welcoming her to talk on plasticity mechanisms and transition from adolescence.
Thank you. Thank you all, and especially those who already heard my talk last night. And here we're going to delve a little bit more into things. And I'm really excited to be presenting to people who predominantly, not all, I know, predominantly are studying aging, because there are, very interesting possibilities looking at overlap, and looking at uniqueness of both of these trajectories. All right, so I'm just gonna go ahead with my. All right, there we go. Yeah, I was told I need a new computer, but you know how it is. You want to buy your students' computers, not me. I'm old, but like it's like email, PowerPoint, and Word. All right, so I apologize for repeating some of the things I said last night, but I really like to make this point very clear that the way that we envision brain maturation during development in broad spectrum is that in the beginning, it is predominantly guided by a process of accumulation. It's getting bigger, you're learning how to walk, you're learning how to talk, etc. Right? So that's typically what we think of development as proceeding in a linear, increasing fashion. However, by the time that you reach adolescence, everything is there. And that linear relationship is now gone. And in fact, you have more, right? You have extrasynaptic connections that will be pruned and, coupling between different regions that will be specialized. So there's a shift in brain maturation to one of specialization. And that is what makes the adolescent period so incredibly important for defining what our life trajectories are going to be. So, during adolescence, then our adult trajectories will be pretty much very influenced and stabilized, and it will position us into aging. And this is something that I've been talking to others during my visit about the possibility of having predictive value by looking at earlier development. And we were talking about that just a couple of hours ago. All right, so what is important about adolescence? It is a time of sensation seeking that is present across societies and across species. And it is normative, it is adaptive. We need these new experiences in order to guide brain maturation, right? But it can lead to risk taking. That can undermine survival and increase mortality increases during this period, which is a period of peak physical health. So there's a concern there. The other major concern, and the reason that the National Institutes of Mental Health have been supporting our work for more than 20 years, is that something is going on, that you see the emergence of psychopathology in adolescence. So individuals who look like they were developing within, normal limits, all of a sudden you see a breakdown. Some of them you can see that the trajectory will go this way, but it's very important to why it is something happening at this time. And the NIMH has made it one of its priorities to understand these developmental mechanisms. They've been asking me for a long time, hey, figure out what these mechanisms are, because then we can inform interventions and we can understand more about etiology. All right, so important for both the decision-making involved in sensation seeking and across psychiatric disorders is the involvement of cognitive and reward systems. And these are exactly the systems that are undergoing very significant maturation in the adolescent period. And now the way that the field has looked at, this relationship between cognition and rewards is that there is a predominant influence of limbic motivational affective reward function over executive functions. It's not an imbalance. This is just a particular balance that characterizes this amazing stage of the lifespan. And we have proposed what's called the driven dual systems model. And what characterizes this model is that we're not looking at the development of executive function as a linear function where adolescents have not reached adult levels. In fact, what we say is that by adolescence, there is access to adult level executive function. It is not reliable, right? It is dependent on context, etc., but there is access to it. And this is important because the type of risk taking that we see, and this is represented in blue in our model. And in the meantime, we know that affective emotional processes are peaking during this time. And what we're suggesting is that because you have access to abstract thinking and planning in adolescence, these are being driven by this hypersensitivity to rewards. And that is what you need adaptively to go and plan new experiences outside of mom and dad, outside of the school, and have that abstract reasoning that will motivate you to get new experiences. And new experiences and sensation seeking are not just substance use and things of that sort. Being interested in something new, like sci-fi or something like that. So we've been trying to understand the brain mechanisms underlying this model. So we've opened up the hood. And today I'm really going to be, more than anything, concentrating on the findings that look at neurotransmitter changes during this time. But I'll give you some of the background of some of your work. And how do we do this? Well, we have a comprehensive battery that looks at cognition, that looks at reward processing, etc. And then we use many different multimodal approaches depending on the question that we're asking. Not just because it's cool, but because we have these questions and we're like, okay, we got to go there. I became interested in dopamine. I'm like, I don't know about that, but we're going to figure this out. And, you make collaborations and you figure that out. The same with MEG. We're like, we need to go beyond the bold response. I've got friends, I'm friendly, as we know. So collaborations are very easy. So we use neuroimaging, to look at structure, function, connectivity, coupling, and neurotransmitters. And we also have really become very much enamored with EEG. And I know that a lot of you are leveraging that as well. All right, so first let me tell you why we propose that cognitive development is occurring in this fashion. And observe also, the variability is decreasing with age, something that many developmental neuroscientists know. All right, so in the past, for a long time, we've been doing these studies, and I'll just briefly, because maybe a lot of you know, we look at inhibitory control by saying, don't look at this light, look the other way. In adolescents, look at the light, and then they look the other way. So it's a very elegant way to tap the integrity of the system. And for working memory, we say, look at this light, come back for a delay period of up to 10 seconds or so, and now command your eyes without any sort of a guide to that remembered location. So you really have to tap into working memory. And we've done this for a very long time. Look at this 2004, blah, blah, blah. We have done longitudinal studies. And this has been very innovative in the sense that we keep thinking that development is linear, and it's not linear. And what we find is what we've been proposing in the dual systems, which is this accelerated improvement through childhood, right? That process of accumulation. And then doing adolescent, deceleration when we think is reflecting specialization after you've accumulated all the basic building blocks. And then there's stabilization. And this has become a very big concept that now is very well accepted, but it wasn't back in the days when we were doing this. But then we were thinking, this is very reliable, sort of results, but it is, based on these neuroscientific approaches of eye movement, etc. Is this really mapping into what we know about cognition as a more overarching concept? So Brendan Turbo Clements, when he was a grad student in my lab, then he went postdoc at Harvard, now he's back in Minnesota. He's like, you know what? Let's do a big data approach. So we took a couple of our longitudinal cohorts, and he brought some other cohorts that were publicly available, so we could make a big data set of more than 10,000 participants and more than 13,000 visits that were involved. And each cohort was assessing cognition in their own particular computer-based assessment. And then it was us with our own test. And the question is, can you bring this together to really understand what is the shape of development and when are we reaching a build-up? And what he found was, in fact, what we had found in the past. This accelerated improvement, deceleration, and stabilization. And as I said last night, I love to make fun of him. It's like, great, dude, you just, proved what we found more than 20 years ago. But this has become very important because, when I've informed the juvenile justice system, that question always arises. Why 18? Is that really a real number? And we're starting to see that on average, a lot of variability, on average, we can start to think that around 18, there is access to stabilization and not only that, but we found that there is a foundational domain general cognitive component that underlies how, no matter how you measure cognition, that you're capturing this. And I know that a lot of you are doing aging work and looking at cognition. And we might be tapping into the same things, and it'd be really great to see that other side of that curve. And this goes very well with what we have been proposing in our work. So what's going on in the brain? And we've been looking at prefrontal cortex. We've done a lot of work. I'm just going to highlight some of the past work. And sorry, there's a delay when I press the button for the next thing. And so for that data set that I told you about that we looked at longitudinally at, it's called the anti-SCOT and the working memory, the memory guide SCOT test. We also obtained fMRI results. By the way, this data is publicly available through NVA at the NIH. Many people have used it and people have published structural stuff, etc. So if you want to, you can always look at it. Anyways, the big point about these first panel over here, these are old studies, was really finding out that when you do a correct executive function response, right, which is less frequent when you're young, but when you do uncorrect it, the way that you use prefrontal cortex is that when you're young, you need to really, really use it at a very high level because of its difficulty. By the time that you reach adolescence, you don't need, it's not that difficult, and this is dorsolateral prefrontal cortex. You're using it just like adults are using it. And we know that DLPFC is very important for making voluntary responses, really telling us by adolescence, you're able to generate these planned voluntary responses. No problem. The problem is that you make a lot more errors, right? So you can't engage the system all the time. It tells us it's not completely mature. And for inhibitory control, it was really the ability to engage the dorsal anterior cingulate cortex, which is the performance monitoring part of the brain, the part of the brain that is very keen to errors and will sound the alarm to engage cognitive systems when an error has occurred. And that mediated and explained the behavioral improvement in inhibitory control. In my lab, I make a big deal about, at the end of the day, what we're trying to understand, the phenomenon of interest, is the change of behavior. We're using brain to try to understand it, right? But the primary thing should always be, linked to what is occurring with behavior. And we found with working memory, it was the ability to engage visual association cortex, which we know from computational approaches is where the actual mnemonic information is kept for visual spatial working memory. Now, another amazing finding that I really like so much is work that we did looking at the variability of the expression of cognitive patterns of whole brain activity, in this case for working memory. And we found that on average, patterns of activity that are used, if you're doing this correctly, are present throughout development. What is changing is the magnitude of the expression of these patterns. And this is something that was really motivated by non-human primate neurophysiological experiments that have found that at the neural and neuropopulation levels, the variability in the degree of activity corresponds to behavioral activity. And that's exactly what we found. But that variability in the expression, sometimes expressed not too much, sometimes expressed a lot, corresponded with behavioral variability in working memory. And both of them were decreasing with age. And I really like that finding because it is suggesting that during adolescence, just like with behavior, the brain is also undergoing exploration and trying things out. Let's try it this way. Okay, let's try it that way until it finds what, based on the outcomes of each time that they're trying, you get to adulthood where like, okay, this is where we need to be. And then all these maturational processes, which I'll talk about with critical theory plasticity, can pretty much cement that this is going to be the dominant way for brains. More recently, we have looked at EEG, and I'm so excited with the work that we've done with EEG. So we looked at working memory during the delay period. A good friend of mine, Earl Miller at MIT, has been looking at this for a long time, has been seeing how important, gamma power and gamma events are for sustaining working memory, which is different from the typical view of working memory where you think, oh, activity is sustained. He says, no, that just happens to look that way because you're averaging across trials. And what's happening in trials are these gamma bursts that are reinstating the memory. We're like, okay, cool. Let's see if that can give us some insight into what's happening in development. And what we found was that with development, you're getting better at working memory, and you need less gamma power and fewer gamma bursts, fewer reinstatements of the working memory. So that is very telling, saying that, oh my God, that transition from adolescence to adulthood really has to do with these maturational processes that are enhancing and optimizing the efficiency of information. So I want to highlight a more recent paper. That last one was last year. This one was this year, and this was led by Tim Calabro, who's my right-hand person. Can't do anything without him. I don't know how he ever did things before he was around. And we were asking this question. We know there are all these important, like synaptic pruning and myelination and specificity of the prefrontal system. We need to understand we've looked at connectivity at great distances, which I'll talk about later. But we really wanted to try to understand the integrity of more of the local functioning aspect of the brain. So we used ReHIL, which maybe some of you are familiar, which allows us to look at the coupling of neighboring molecules in a particular region, right? So we can assess how effective this coupling is. And one would think, hey, with development, there's probably greater coupling, as you get older, and that's how the brain works. So we looked at resting state. We also had them do a memory guided saccade test. And here we're showing you how we did the coupling. We looked at the cortical ribbon, right, across the cortex and looking at these neighboring voxels. Of course, each voxel corresponds to many, 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 many neurons. So we're not talking neuron in particular. But what did we find? You would think increases in coupling. But that is not what we found. We found decreases in the coupling of neighboring voxels. In other words, these neighboring areas with development, as you're getting better working memory, are able to work independently. And this is in resting state. And it is associated with working memory. The more independent these different localized regions show, the better that you're able to engage executive function. So what could that be reflective of? And it could be that this independence allows better dimensionality of processing complex information.
Are you willing to take
Go for it. Oh, that interesting. I'll get my drink. Yes.
somewhat nerdy question? Maybe it's just the right hand to answer.
Yeah.
So if you're looking at neighboring voxels, take a look at the spread function. Any two voxels that are close to each other, their correlation is simply so I'm wondering whether this reduction development vascular system not. So if it's the case that specificity of the whole signal to the vascular bed increases age, it's exactly. So it could be that mediated this is some change in vascular system. It's leading to spread function, function.
No, that's an excellent question and very reminds us of one of the reviews that we got. I think it was that you mentioned. Where we had to do additional analyses to really prove that it was not due to that. And I can't give you the details because I'm not the mastermind with that. That said, I think that you do have a very important point. That there could be just differences in the vascular aspect, and that's something that's come up a lot from my career. And we don't believe that it is so, and there is some evidence that maybe that is not the case, but it could be the case, right? So that is something to keep in mind. So what he did to see, is this really buying you anything to have this independence? So he looked at effective dimensionality. And that is exactly what he found, that there was a correlation between the independence of these neighboring voxels and increases in effective dimensionality, really meaning that this independence is really correlating with more effective processing of complex systems. And what really, nailed it down for us and was the fact that we have a close collaborator, in Vanderbilt, who does very similar work that we do, but on monkeys. And this paper came out after ours in Interneurope, and we're parts of it as well. Really? I thought what? Sorry. And in fact, that is exactly what they find with neurophysiology, that this, effective dimensionality is increasing with development. All right, so now I've really, given some compelling evidence, I think, that the locus circuitry specializes into adulthood, resulting in independent processing that supports this complex information, encoding leading to mature executive function. But we were wondering, are there parallel changes in the temporal domain? And this is probably the newest information that I'll be sharing with you today. So please be kind, as I'm still trying to wrap my head around it. I have the paper here for me to read. I've read other versions of it. So the idea is the following, that there are these intrinsic neural timescales that you can capture with EEG. And you can look at this with autocorrelation windows. And really what you're doing is that you're looking at this time series, doing rest and doing task, and seeing how predictive that time series is of itself. And you can see that higher correlations will be reflective of longer autocorrelation windows. And why do we care about these autocorrelation windows? It's because we want to know about the efficiency of the speed of processing and how that is changing with development. So what we did in this paper that we're really shooting high, I'm starting to realize, I could have known this. We're always like, oh, let's go to Nature Neuro, let's get to Nature. No. Holy cow, how am I going to pay for this? Yeah, we have to rethink this model. Anyways, so I continue to talk about this one population, by the way, of subjects that we've tested in a million different ways, and I'm highlighting almost everything that I'm showing you, all the new works from this one population of more than 160 individuals, 1032. It's a longitudinal study with G and 7T. It's very good. Anyway, so here for this autocorrelation window, we interrogated what we found with G during resting state and during our working test. We also have a collaborator with a neurosurgeon, and I know, it's Noah. Where's Noah?
Teacher.
Oh, yeah, that's what she did tell. Yeah, we've been talking about how we need to get all of these, SEEG studies together and make a database. Because here we have 14, but it is SEEG. So we wanted to see, are we going to see the same differences in the properties that we see with the surface EEG? And then, working with Christos, we were able to interrogate this in the development of. And what we found was that the autocorrelation window decreases with development. So you have much more speeded, information processing. And the window is actually slower when you're doing a working memory test. It makes complete sense. But that distinction is really predominant in adulthood. In adolescence, they're slow regardless. That is their base function, to be processing in a more slower function. And this is an area where I think aging might be similar, but it might have different benefits. So I think I'm just thinking off the top of my head that in aging, slower might be good. And maybe in adolescence, slower might be a reflection of limitations. I know I'm getting slower and I'm And we found this shortening of these windows across development were replicating in SEG and in CAT LM. So now we're really feeling that this is something that we think is also giving us a view of how optimization is occurring through development. And we saw that the shorter windows are associated with improved working memory and with EEG indices of EI balance, which I will tell you about in a little bit, which I'm sure that you know, I think I talked to Nick and other people that you guys are interested in this aspect of EEG. Yes, breakfast. Yes, I remember that. Kim, yeah. Anyway, so, and that we see, which I'll also tell you about as well, that is these windows are shortening, they correspond with changes that we're seeing in intracortical myelination of the deep layers. And I'll explain this to you because there's a whole story about plasticity. But in other words, this autocorrelation window is decreasing in development. So from adolescence to adulthood, spatial and temporal specialization of neural circuits and prefrontal cortex support complex computations underlying the development of executive function. So that really has led us for a long time to thinking about plasticity in the brain. We know is available throughout the lifespan, but even more about critical period plasticity. So people talk about adolescence as a sensitive period, blah, blah, blah. And I had these discussions with Bart Larson when he was a grad student. He's in Minnesota now as well. And I was like, really sensitive period? What does that even mean? We know the mechanisms for critical period plasticity. Let's see if that exists in the adolescent period. So just a little reminder, what we know about visual cortex, or from Takao Henton as well. You're born, you open your eyes, you get all this influx of light, which increases glutamatergic excitatory function and breaks the balance with inhibitory GABAergic function. That balance is broken. That is a trigger for that part of the cortex to reorganize so they can now, you know, so the neural architecture can now process this very intense high signal information and then go back to balance, in this case, early in development by the maturation of parallel human inhibitory neurons. And that's how they have found that that's what happens. So that balance, we're like, okay, let's see if that exists. And what Bart did here is that he looked throughout the literature, postmortem studies, humans and monkeys, and found that in fact there are aspects of glutamate that are decreasing through the adolescent period, which is huge, because really before this people are thinking, oh, nothing changes. Yeah, there are important changes. And that could be related to synaptic pruning, which we know is happening. And there are aspects of GABA function that are increasing. GABA is very complex, right? And it's really, you know, kind of like specific to parvalbumin varicosities that are increasing. Other aspects of GABA are doing. But that was enough to write a grant. And by the way, this paper just came out. It's another review paper that really delves into all the aspects of critical period plasticity, really integrating the human and animal studies. I'm honored to be an author because it was really driven by Val Sitner, who's this amazing postdoc. And it really talks about the timing of critical periods. Anyway, so we were thinking, you know, with Barbara, like, okay, let's write, you know, a grant about this, but how can we look at GABA? And we started talking to experts in the field, and the then director of the MR Center, who left, and now I'm the freaking director. And he's a physicist, I'm not, but you know, but in any case, so, and he said, if you want to look at GABA, and you, sorry that this is so small, but you can see with spectroscopy the signals of different metabolites. And GABA is very, 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 very tiny. And it can be buried under the macromolecules that, he argues cannot be resolved at 3. You have to more than double the signal to noise ratio. We're like, okay, let's go there. And we use not just regular spectroscopy, but magnetic resonance spectroscopic imaging, which provides you with small little voxels and a whole slice, so you can look at several regions at once. So, and it involves all the shimmying, a special foil. But there are new ways. I think the new Terra X, which we got, but things have happened, and hopefully they'll be working in a few months. So that's what we did, and we looked at different parts of prefrontal cortex, and we had hypothesized here. And by the way, most of my graphs have age horizontal axis, and what I'm showing you here are the changes that we saw in left and right DLPFC, so almost like a repetition of glutamate in blue and GABA in red. And we had hypothesized that it would decrease, that the glutamate would decrease, and that is what we found. We had hypothesized the GABA would increase, and that is not what we found. As I said last night, I love it when hypotheses are wrong, because that really forces you to rethink how we think about this. And what we think is occurring is one of two things: maybe the maturation of these aspects of inhibitory function, like in visual cortex, have already matured, or, and possibly more likely, our comprehensive, measure of GABA is not sensitive enough to identify the aspects of inhibitory function that are probably still mature. But what we really cared about is this balance changing. And that is what we find in different parts of prefrontal cortex, so that you have this, too much excitation that then is recovered because of the decrease in excitation to increase balance. And we published this, and we're super excited, blah, 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 blah. But we need more evidence than that. And we've gone into this pattern in the lab where we want to find evidence in a bunch of different modalities to make sure. So that's what we did on the same subjects. Oh yeah, you guys don't see the real. Okay, there we go. All right. We looked at it. So this was led by Shane Kuhn, a BioE PhD student of mine who is now a postdoc. She is absolutely awesome. She's very BioE. She's like, oh yeah, I think we'll do this, and this is what we've done. I'm like, oh my God, you found that? Yeah, it's hilarious. Anyway, so she found that there's this approach in. Raise your hand if you've heard of FOOF or spectrogram. So I encourage it. So it's a way to look at the aperiodic aspect of the EEG signal, right? Not the periodic, the aperiodic function. And you can look at the offset and the exponent. And what has been found in the past with younger children is that it corresponds to the changes in EI balance during visual, cortical, critical period plasticity. So we were like, okay, we've got this information on the same subjects who did the 70 MRSI study. Let's see what it looks like. And that is exactly what we found. A decrease in the exponent through adolescence in frontal cortex, which is super exciting. I mean, it looks so simple, blah, blah, blah. But I mean, this is really a concept that people haven't thought about, that in adolescence you can have evidence for such, significant changes. And it's like ahead of time. And we saw that there was an incredible association between the EEG evidence of changes in EI balance within the same subjects with the GABA glutamate changes in EI balance. So boom. And the changes in these metabolites were mediating the changes that we've seen in the study. So with EI balance, you would say, okay, now the brain can really improve the signal to noise ratio. So on the same subjects, we did also that. There we go. So what we did in the same subjects, we did an auditory steady state response test, where you hear these different trains of clicks, including over to the gamma oscillations. And you can derive SNR because you can get the total power of everything that's occurring during the session. And then you can subtract the evoked power because you know all the click trains. And that gives you the spontaneous power. And SNR can be seen on oh my, sorry guys, I'm getting to Microsoft updated system. It's not helpful. Anyways, and we can get a measure of signal to noise ratio by looking at the ratio of evoked to spontaneous. And what do we find? I'm just going to go ahead. Press ahead of time. We find that evoked responses, although it looks like it's going down, it is not significant, but spontaneous power is decreasing significantly with development that results in increases in signal to noise ratio. I was not having these problems last night, but that's all I have to say. And we see that the increases in SNR are associated with the changes in glutamate and GABA balance that we found in the same subjects. And it is as SNR is improving, so is working memory. And this is already controlled for 8, so that we see greater SNR, greater working memory. All right, so this is really telling us, there's important changes that are occurring that are shifting that EI balance that tells us about critical period plasticity. Another very important aspect of plasticity are closing factors, myelination. And we've looked at that, and many others have looked at that, by looking at TI studies. Here, this is an old study. We looked at diffusion spectral imaging. You can see in this heat map, periods of growth or myelination is growing and so forth. But what we found that what we did that was a little bit different is that we looked at what's called the term regional terminal zones. When white matter is reaching the gray matter, we saw that that has continued myelination. So that's been percolating for a long time. So in comes Val, this brilliant postdoc, and she's like, you know what, we can leverage this 7T data by looking at the quantitative R1 measures that are available there. And that is a more direct, reflection of myelination. And we can start to interrogate different depths of the cortical ribbon. We're talking about just the gray matter, not the white matter. And start to think about what we know the different layers of the cortex are doing. So superficial layers provide intercortical connectivity that is needed for learning, etc. And the bottom layers of the cortex are providing top-down connectivity, which is what you see predominantly changing through the adolescent period. So we wanted to interrogate these things, and this is under consideration in Nature. That's going to be pricey. Just gone to my journal, Development of Cognitive Neuroscience. But people complain, oh, $3,500. Yeah, it's not, 10,000 or whatever. All right, so this is what she has been finding, that the superficial layers, which I find fascinating, have this linear and continuous reflection of plasticity, which is great. You guys do aging. It's probably still there. It's what you're doing as we're speaking right now. And maybe some things are like, oh, I didn't think about it that way. That is possibly supported by the superficial layers. The deep layers, though, are showing overall in frontal cortex what I told you before: acceleration, deceleration, and stabilization from adolescence to adulthood. And what she has done even further, because Val is very much known about sensory motor association axis. We got the spinning wheel. So she's also looking at hierarchical differentiation of different processes. And what she found that in, the most posterior aspect of frontal cortex, you see that all the layers are showing this early maturation. When you go to DLPFC, there's this differentiation of the deeper layers of top-down connectivity showing this adolescent maturation. And then the anterior cingulate cortex, which I've told you about before, across the different layers, it's showing this persistent plasticity across development, which I think is very telling, which I think might have implications to aging. You look at cognition, they have sensitivity to error processing. I'm just saying. And what she has found is that these changes in these top-down myelination of the deep layers correspond to the changes in EI balance that we found. And it also corresponds with a two-stage learning task with learning rates and accuracy when you look at the association between the intracortical myelination. So again, anchoring it back to. So what we think together is it changes in GABA glue, in EG, intracortical myelination, really suggests critical period plasticity through adolescence, which is huge. I mean, all right. And, this very much supports our driven dual systems model, that there is a particular curvilinear change during this period. All right. So now for the second part, and let me know if I'm
5 to 8 minutes.
Oh, 5 to 8 minutes. Okay, I'll hurry up. The limbic part of the brain. Sorry, I get so into it sometimes. It's also undergoing its own unique maturation. And we know that there is this heightened reactivity to rewards that we have found in the past that even assist with inhibitory control if that is dependent on obtaining a reward. And this has led us to really realize that there's something very critical occurring in the ventral striatum and the dopaminergic system. So one thing that I really want to show you, because it might be related to some of the work that you guys are doing, is that we did this study in a PET MR machine, get both things at the same time, and we looked at tissue iron. This tissue iron is a necessary step of the production of dopamine. It is predominant in the basal ganglia. It has been studied for a long time in aging, in Parkinson's, and other dopaminergic related disorders. And we also did PET in 18 to 30 because it's difficult to do PET on pediatric populations. And what we found was that tissue iron is increasing with age, and it is particularly associated with direct PET markers of dopamine availability. And that is huge because this is a non-invasive way to look at a very important aspect of dopamine, which I think is very important in aging. And even though here we looked in this study, we looked at a direct measure of tissue iron, you can obtain this tissue iron measure from resting state or task fMRI. You just time average the T2 star that you already have there. And we collaborate with so many people. They're like, how do you do it? And we'll have a very comprehensive paper that will be submitted, hopefully in the next couple of weeks, that really tells us how to really be able to enact this. And it's been very revealing. And another really interesting finding that we found here when we looked at 18 to 30-year-olds who got the PET as well, is that the amount of dopamine starting to show a period of stabilization from 18 to 30. But receptor D2, D3 receptor density in the striatum is something akin to synaptic pruning, which leads us to think that perhaps developmentally, how much dopamine you're going to have might be determined, obviously, with genetic disposition. But by adolescence, it's like, okay, this is how much dopamine we'll be operating with. Now we can go and determine how many receptors we're going to need to have that perfect match to get the system going. And I'll skip around a little bit. See. We find evidence that dopamine is very much involved in these changes in GABA glue, but I did want to get to substance use disorders, just one particular aspect of it, which is in a big data set, we have found, not surprisingly, that teenagers are trying out substances. There are different sorts of trajectories that occur. One that's very well known, a peak in substance use, adolescents and young adults, in college and so forth, will try substances, but they will not persist. But what we have found that's very interesting, one is that measures of impulsivity and measures of inhibitory control are associated with substance use. The really revealing one that was one of those aha moments is that it turns out that those with the least amount of tissue iron, right, which is reflecting dopamine availability, are the ones that we see are using substances and have higher impulsivity, higher risk taking. How could that be the case? And I will finish with my latest model that I'm proposing, which is you have presynaptic dopamine, and I'm showing it here in blue. It looks very much like the driven dual systems, by the way. And what animal studies have found, and what a lot of our human studies have found, is that presynaptic dopamine availability is, developing in that similar fashion as cognition: acceleration, deceleration, stabilization. But postsynaptically, you have a peak that then decreases. So what could be happening when it comes to risk taking? Because you wouldn't think less dopamine, more risk taking. And this is what we're thinking. When you're young, you don't have a lot of presynaptic dopamine, but you have a lot of transmitters. And getting that system going is difficult, right? And that maybe risk taking provides a compensatory, mechanistic way to get the system going. As you get older, you have more dopamine, you have fewer receptors, and now you have a very nice match that you no longer need those compensatory. All right, so I'm just going to go ahead and go to the end. We have looked at habit, which is very cool, and we're able to see that habit is increasing with development, and it can be used to predict alcohol use into adulthood. And I just want to get to the end. Just have to spin. Just want to show. Where's Chris?
I suspect your machine is updating.
The what is?
System is updating.
Yeah, we had to look. Spinning wheel.
Guessing that's what's happening.
Yeah, I have this. I'm sure some. It's kind of funny. As my. All right, so I'll start telling you about the conclusion. So what we're thinking is occurring is that adolescence is a very unique period where there's a lot of plasticity, and there are a lot of changes that are occurring that might underlie why we're getting psychopathology at that time, right? It might have been a predisposition, but now it's finally coming to the fore. But it's also an opportunity to affect trajectories. And that's what we really want to concentrate. when, we're thinking about these developmental things. So I'm just gonna go ahead and open it to questions because
Hopefully
this is not working.
Yeah,
Yeah.
I'm sorry.
Yeah, so in the meantime, we can,
So we will pause and wait for your slides to catch up with you and have some questions.
Yeah,
Okay. But first
exactly.
off, thank you so much. We are open for questions.
Great talk, thank you. I actually just started getting my questions very quick, but in terms of it's, trajectories, do you see that in data? Just like we get variance in a lot of these measures, do you see differences at the extent of this positive?
That's something that we're trying to investigate. The thing is that we've been really, our cohorts are normative. So we do not include anyone who has any sort of psychopathology in themselves or first degree relatives, which by the way, it's becoming more difficult with time. And it's like, who are these people to begin with? But we still have a lot of variability, for example, in indices of adversity as like the ADI and neighborhood and so forth. So we're trying to map and see, because we know from the literature that stress can and has been shown to affect the timing of critical period plasticity. So the data is there, and there are a lot of people that are interested in pursuing that. And we also have assessments of stress and stress perception that I think will end up with what your question is leading to. That, there is going to be variability that is very telling as to what can affect plasticity.
I'm curious if you're aware of a proposed biochemical mechanism for the relationship between tissue iron and dopamine availability.
So it's actually something called ferritin, and it is I'm not a molecular person, but in the making of catecholamines, you need this piece of the recipe that will eventually, end up being dopamine or epinephrine. It's also involved in myelination. And one might ask, well, then how do you know it's dopamine, right? For a couple of reasons. One, it is, when you look at this, if you go and you start looking at the data, the basal ganglia will, it's the complete hotspot.
Yeah.
And the one that's reliable and that you think about it. There's also been, direct monkey studies that have shown the relationship between these measures of tissue iron in the basal ganglia corresponding to these sorts of mechanisms. It's a very good question to ask, but it's new and it's very cool. Oh,
So one more.
Well, definitely.
Keep going.
Yeah.
Yeah, I mean, you have this, if you're doing FRI, you have this data.
What is your measure of dopamine availability?
It's that tissue iron.
Okay.
So what we did in that PET study,
Yeah.
we're like, because like I said, a lot of other people have looked at this and they would say, oh, the integrity of neurophysiology, right?
Yeah.
And I'm like, no, is it dopamine? So what we did is that we obtained that tissue iron, right, in adults, at the same time that we obtained DTBC with PET, which is a measure that tags VMAT. So it gives us a measure of dopamine availability.
Okay.
And raclopride, which gives you a measure of the density of D2, D3.
Okay.
Yeah, no, it's a great question because I speed it through.
Yeah. Thank you.
I have a question on your MRSI data. I find it very fascinating. And I was wondering if you find differences in like the prefrontal cortex or certain regions that you're seeing that like a massive imbalance versus more of a stability even within the prefrontal cortex, because you have the ability to localize that.
Yeah, that is a really good question. And, I'm here telling the story, that's a bigger one, blah, blah, blah. And you can see it in the paper. For example, the insula seems to be doing something different. And it seems to have perhaps a longer critical period than other areas. Now, we had a slice that hit a lot of prefrontal cortex, but also some thalamus.
Okay.
Yeah, and I think a little bit of the caudate, and then it went into visual cortex. but signal to noise ratio there wasn't too good to look at. But we are very interested in looking at that, and we also had another slice that looks at the hippocampus, and we're finding very similar results. This, glutamate going down, changing the balance in the hippocampus, and associated with all these indices of learning and so forth, which will be the next slide. Tell you guys about that.
Are there any questions online?
There
Yeah.
are questions online.
Sorry, I'll come back to you. Sorry, say again?
No questions online.
Yeah, so learning all your part of your job, like during childhood, there is a rapid growth. And if I understand correctly, during adolescence, addition, being safe. So some people childhood is critical.
That what?
Childhood could be more critical in terms of intervention for promoting cognitive perspective. So I was curious about your thoughts on how the intervention of the future of child and adolescents given their random.
Yeah. I mean, that one postdoc Val is she put in a K99, really leveraging different periods and types of critical period plasticity. So, for example, doesn't like me.
Saw Mick really quickly.
Yeah, so that there, for example, the cortical connectivity, which is something she wrote. Okay. Ahead of time is really showing that there's, as you're suggesting, there are different plasticity at different levels. And I agree with you that, given that accelerated growth in childhood, that would be, I've got work to do after this. That it'll be different and unique, which is, part of what I've been saying for a long time, that it's very unique and different from childhood. But there's a lot of work to be done there. You're welcome to look at our data.
I'm going to bring this formal side of things to a conclusion. Are you willing to hang around and take
Yeah.
comments, questions after? So can we just say once again, thanks for such a